When neurons communicate, they release chemical messengers across the synaptic cleft to trigger a response in the receiving cell. In the human central nervous system, two specific amino acids handle the bulk of the inhibitory signaling: Gamma-Aminobutyric acid, or GABA, and Glycine. This table categorizes these two transmitters as inhibitory, but if we check the fine print at the bottom of the table, we find a critical condition. The postsynaptic effect, whether a cell is excited or inhibited, depends entirely on the nature of the receptors and ion channels activated by the transmitter. A neurotransmitter molecule does not possess an inherent ability to inhibit a signal. Inhibition is an emergent property. It is the result of specific receptor architecture and precise electrochemical gradients maintained by the receiving cell. Recognizing this state as conditional is the first step to understanding how subtle metabolic failures can completely invert the function of our neural networks. The brain synthesizes GABA by pulling glucose through the tricarboxylic acid cycle, metabolizing it into glutamate. The final conversion to GABA requires the enzyme GAD. GAD cannot operate alone. It requires a specific cofactor, pyridoxal phosphate, which is derived from vitamin B6. Without adequate vitamin B6, the entire synthesis pathway halts. When an early infant formula omitted this vitamin, infants suffered severe, sometimes fatal seizures because their brains physically lacked the GABA required to stop unchecked electrical activity. Glycine synthesis follows a different route, utilizing the amino acid serine as a precursor. Once synthesized, both GABA and glycine rely on the exact same vesicular inhibitory amino acid transporter, or VAT, to load into synaptic vesicles. If a presynaptic neuron cannot complete these specific metabolic conversions or load the resulting molecules, the chemical off switch is absent. The network will overexcite before the receiving cell even plays a role. When the chemical supply is intact, the molecules cross the cleft to bind with their primary targets on the postsynaptic membrane, inotropic receptors. These receptors, for both GABA and glycine, are constructed as pentamers. 5 separate protein subunits combine to form 2 distinct regions: an upper extracellular domain that protrudes into the cleft and a lower transmembrane domain embedded in the cell. Viewed from above, the alignment of these 5 subunits forms a central pore. A ring of positive charges lines this internal channel, creating a highly selective gateway specifically for negatively charged chloride ions. At rest, the channel is closed. When a ligand binds to the extracellular domain, it triggers a physical shift in the protein structure. The central radius expands from 1.4 angstroms to 4.4 angstroms, creating enough clearance for hydrated chloride to pass through. This mechanical interface can be exploited. Drugs like barbiturates bind directly to the extracellular subunits of the GABA-A receptor. They enhance the receptor's function, artificially holding the pore open to depress the central nervous system. Conversely, the plant alkaloid strychnine achieves a lethal effect by binding to the glycine receptor. Instead of opening the pore, it locks the channel shut, completely blocking the influx of chloride and triggering fatal convulsions. The straightforward mechanical design of ionotropic channels makes them highly efficient at halting electrical signals, but it is exactly this simplicity that leaves them highly vulnerable to chemical hijacking. The brain also employs a slower inhibitory system, using metabotropic GABA-B receptors. Unlike the pentamers, these are heterodimers, requiring both a B1 and B2 subunit. Activation begins when GABA binds to the B1 subunit, aptly named the venous flytrap domain. This receptor does not contain a chloride pore. Instead, closing the flytrap domain activates intracellular G proteins. These downstream messengers then execute the inhibitory command by opening potassium channels and blocking calcium channels. Together, these 2 receptor types provide a complete regulatory system. Ionotropic pores act as immediate, localized breaks, while metabotropic receptors provide broad, sustained modulation across the network. Because primary GABA and glycine receptors operate merely as open pores, they cannot actively push ions. The direction chloride moves is dictated solely by the cell's electrochemical driving force. In a mature neuron, cotransporters eject chloride across the membrane, maintaining a low internal concentration. Because this level is low, chloride's equilibrium potential sits below the resting voltage. When the receptor opens, chloride rushes in, driving the voltage down, inhibiting the cell. During development or pathology, a different transporter dominates, pumping chloride into the neuron, creating a dense buildup. This massive concentration forces chloride's equilibrium potential above the resting voltage. When GABA binds, pressure forces chloride out, spiking the voltage upward, firing the neuron. Metabolic disruptions can force mature neurons back into this high chloride state. When a brain is starved of energy, the gradient degrades, and the very chemical responsible for calming the network begins actively exciting it. This mechanical inversion is implicated in severe pathologies, including extreme network disruptions seen in anorexia nervosa. It proves that neurological inhibition relies on a fragile electrochemical balance, easily dismantled by the environment outside the synapse.